So, immunological surveillance. This is where our NK cells come in. Our NK cells are our lymphocytes. Most lymphocytes give us specific defense, right? Our B cells and our T cells are specific, adaptive defenses. NK cells are just different. Our NK cells are constantly roaming around the periphery, monitoring, looking for anything that's abnormal. And then when they see anything abnormal in the tissues, they'll try and destroy it. So when we look at the NK cells, we see that they can respond more quickly than T cells and B cells because they don't have to be activated. Remember when we looked at the T cells and B cells, you need the antigen presentation to occur. And then you've got all of this mitosis and differentiation, and you produce antibodies, and it takes time. That's a really nice big response that you end up with, but it takes time to happen. The NK cells don't have to be activated. They're not specific against any specific antigen. They see something abnormal and they try and destroy it. They're just natural killers. So they are quicker. Again, it's not this nice long memory response that gives us long-lasting immunity, but it's fast to try and take care of something right off the bat or really quickly. They can attack anything. So they can attack bacteria. They can attack foreign cells, foreign or like transplanted cells. Their kind of claim to fame or what they're really important for is our cells that become abnormal in some way. So our body cells that become cancerous cells, or our body cells that are infected with viruses, those are our cells that become odd, or they're unhealthy in some way. So the antigens they display are going to be abnormal antigens. And that's when we really rely on the NK cells, or natural killer cells, to come destroy them. So how do they do that? Natural killer cells work by going and binding to the target cell. Okay, so we'll see that they adhere to their target cell, whatever they want to destroy, and then they release perforins, and some sources will say also granzymes. Okay, so definitely perforins. Granzymes are kind of a, why are they doing this? We're still trying to figure it out thing. So perforins perforate. They perforate the target membrane. They travel across to the target cell and they create a little pore or a little hole in the plasma membrane of the target cell. That has a couple different functions. One, that can kill the cell, right? You need your plasma membrane to be intact and working. If you just have holes all over your plasma membrane, it doesn't work anymore, and the cell will die, right? The cell will burst. It can't function any longer without a plasma membrane. Also, these pores can allow for the granzymes to enter into the cell. So we think that that's extremely important in virus or cells that are infected with viruses. It's important to create pores for the granzymes to enter, and then we think that those granzymes help us out to help inactivate the viruses and induce that cell death or apoptosis in the target cell. We're going to take a second and look at how this happens. So NK cells, remember they're lymphocytes, but they're actually responsible for non-specific defenses or innate defenses. So notice a few things here. This is the NK cell. This is the abnormal cell. So this can be one of your cells that's got virus in it. It can be one of your cells that become cancerous. And what we mean by that is that the cancerous cell essentially loses cellular control. It should divide at a very controlled rate. As it develops, it should be able to differentiate into a nice, specific, functioning cell. Once it gets too old, it should die. Like, that's what's supposed to happen. Some cells lose that ability though, and they just start dividing and dividing and dividing, and they're dividing so fast that they can't differentiate. They don't have time to actually develop the way they're supposed to, so you just get a mass of really non-specific, non-functioning cells. They don't die when they're supposed to. They're not working right, but they lose that ability to die. So they just keep going, and they take over all of the tissue that's surrounding them, and that's a cancerous cell. So this is some abnormal cell. It can be cancerous, it can have a virus, whatever. This is our NK cell. Notice where the Golgi apparatus is. Remember, the Golgi apparatus is the packaging plant, right? So if I make any kind of nasty chemical, like a porphyrin or a granzyme, I don't want that just floating around my NK cell. right, or that can damage the cell. I have to package it in a protective little vesicle. The Golgi does that, right? That's our packaging plant. So notice the Golgi apparatus is over here. Okay, that's where it's packaging up all of our nasty little chemicals inside these vesicles. Here you see that it's adhering to the target cell. Very important, right? That the NK cell adheres to this abnormal cell. You don't want to just releasing nasty chemicals all over the place. It needs to release them right at this target cell. So you need it really close. Now notice the Golgi apparatus. Look at it here, look at it here. The Golgi apparatus moves. So the Golgi will move until it's facing the target cell, because we want these porphyrins and granzymes to be released directly across from the abnormal cell. We do not want them to be released over here. If they're released over here, they can go to this cell, which is healthy, or this cell, or this cell. We don't want that to happen. We want them to only go to that abnormal target cell. So the Golgi rotates towards the target cell. So you see it realigning towards the abnormal cell. And then it secretes the porphyrins. This picture's from an old text that just talks about porphyrins. The newest edition talks about granzymes as well. Okay, so again, it's just kind of, we're figuring out the granzyme thing now. But we've released porphyrins and granzymes. Here you see again that this is a very, very close proximity to the target because it's adhered, it's actually attached to it. So these vesicles, when they merge with the plasma membrane and we release these porphyrins, they do not have to travel very far, right, to make sure that they just go to that target cell. Here you can see the porphyrin molecules coming together and combining, right, and they end up making this nice pore, right, a nice pore, a nice hole in the plasma membrane. That itself can destroy a cell. Again, we also said that granzymes can be present, and these pores can create little holes for the granzymes to enter. And again, we're thinking that that's super important with virus-infected cells. Then that's NK cells, porphyrins, perforate the plasma membrane. And then here you see the target cell is lysed. So interferons. Interferons are extremely important in the fight against viruses. So let's just take a second to talk about viruses and how viruses work, because that's going to be important to understanding interferons. So what happens with the virus is, here's my cell. Okay, and remember my cell, I have like my nucleus with my DNA, and I've got these ribosomes, right? And the ribosomes are where I make proteins. So when you look at a virus, the virus does not have the ability to replicate on its own. A virus is not a full cell. A virus is essentially just genetic material. It cannot replicate on its own. It has to use your cell to replicate, kind of like a parasite lives off of you. Right? The virus has to utilize your cells in order to make new viruses and spread. So what happens is the virus will gain entry into your cell, and then it takes over your cellular machinery. It'll utilize your cellular machinery to replicate, since it can't do it on its own. It uses your enzymes, it uses your organelles, and it replicates. So he comes in here and he makes more and more and more and more and more copies of himself. And then when he's made tons of copies of himself, he kills the cell, right? When this cell bursts, all of a sudden, all these viruses are free, right? And they'll all go out to all the neighboring cells and infect them. And that's how the virus spreads, okay? It enters your cell, it replicates, it kills the cell, and then it spreads over and over and over again. So interferons are proteins that are really important in the fight against viruses. Interferons can come from our lymphocytes. So our T cells and our B cells, once they're activated, some of those chemicals that they'll release are interferons. Interferons are also released from virus-infected cells. So your body cells, once they get infected from a virus, will start releasing interferons. The interferons are not going to help them at all, but they'll help the neighboring cells. It's kind of a, okay, it's too late for me, but maybe I can help the guys around me signal. Okay, so what interferons do is the interferon interferons go to neighboring cells, so chemical messengers that trigger the production of antiviral proteins in healthy cells. So they go to all of the neighboring cells and they say, hey, look, there's a virus that's nearby. You need to start making these antiviral proteins. Like you need to protect yourself against this virus that's in the area. So these antiviral proteins, what they do, they don't actually kill the virus. They can't stop it from getting into the cell. They don't actually get rid of it, but they stop it from replicating. Okay, so all of those neighboring cells, all of these cells here, the virus can get in them, but that's it. And that's really not a big deal. It can't replicate, so it can't keep spreading. So the antiviral proteins will slow the spread of infection. And then we rely on our other immune system cells to come in and engulf and destroy these cells to completely eradicate the infection. Right? A macrophage can come and engulf this, or we just looked at NK cells. Right? Remember we said NK cells are really important for looking for our cells that aren't quite right? So this is our body cell that's not quite right because of this virus. The NK cell can come in and destroy it. Okay, so let's talk through that again. So I have a cell. This cell gets infected with a virus. Okay, the virus starts replicating. This cell knows something is wrong, right? It's got a virus replicating in it, so it releases interferons. Okay, so it releases all of these interferons and they travel to the neighboring cells. They tell the neighboring cells to start making antiviral proteins. So now, antiviral. All of these neighboring cells have now been warned and they've all made antiviral proteins. Eventually this cell is killed, right, and all the viruses get released. Now the virus comes and it enters in here. The virus comes and enters, the virus comes and enters. But because of these antiviral proteins, it cannot replicate. Can't. So it stops spreading. It stops that cycle of replication and spread, replication and spread, replication and spread. Then of course we'll call NK cells, macrophages to the area, they'll come get rid of it, get rid of it, get rid of it. But we've stopped that massive spread of the virus because of these interferons. Is that okay? The complement system. So we have 11 complement proteins in the plasma. Okay, so this is a specific type of plasma protein, the complement proteins. Just like all the other proteins, we make them where? In the liver. So the liver makes these 11 complement proteins. And these proteins together form the complement system. One of the major functions of the complement system is to help antibodies. Okay, so they assist antibodies, they work together with antibodies in the destruction of pathogens. That's where this name complement comes from. It complements the action of antibodies. It works with them to enhance the action of our antibodies. There are actually a couple different functions though. We'll see how they work with antibodies. They cause histamine release. They can form something called a membrane attack complex. They do lots of different things. But one of their major functions is to help antibodies. So when we look at the complement system, it involves multiple pathways that kind of come and activate these proteins in cascade type reactions. So like clotting. Remember with the clotting cascade, we had all these clotting factors, and you have to activate one, and then that activates the next one, and then that product activates the next one. Right? And you have this whole cascade reaction that occurs. The complement system works in kind of the same way. We've got all of these cascade reactions, and again, we have 3 pathways. Luckily, you don't have to do any memorization this time, like with the clotting cascade. But we do have 3 different pathways, or 3 different ways that we can activate this complement system. We've got the classical pathway, This is not alternative, this is alternate. Alternative path, it's like a left side. The alternate pathway and then the lectin pathway. The point here though is that all three pathways, no matter how we start, they all end with the activation of C3. C3 is just complement 3. Complement system, complement proteins, C3 is complement 3. We split complement 3 into C3A and C3B. Those are the active versions. That's what kind of gets us going to activate the rest of these. So what are the things that we can do? We can form something called the membrane, oh here I have it, the membrane attack complex. So once we activate C3A and C3B, they join together with numerous other proteins and they form the MAC, or the membrane attack complex. It does just what it says. It attacks the plasma membrane of the target. So kind of like the porphyrins, how they create little pores, kind of like that, right? We destroy the integrity of the target's plasma membrane. That would destroy the target cell. We mentioned that they help antibodies. So when we look at the complement proteins, what we'll see is that we have antibodies binding to targets and we've got complement coming onto the antibodies, and that enhances the activity of the antibodies. We call this opsonization, but essentially it's like kind of like flashing neon lights, like, hey, this is something that's marked, this is something that's bad, you need to come phagocytize it right now. And then I think I used the example of buttering up the bread, right? That it makes it easier to swallow, easier to eat. This opsonization makes it easier for phagocytes to engulf targets. So it's like flashing red lights to call phagocytes to the area when you combine complement proteins and antibodies, and then it butters up the bread. It makes it easier for them to be engulfed because of that combination. It also stimulates histamine release. What does histamine cause? Inflammation, right? So that increases inflammation. Increasing inflammation, again, we said is protective, right? In a second, we'll talk about how it helps to limit the spread of infection. And then it also brings more white blood cells to the area so that they can help fight whatever the issue is. All right, the last couple things or the last couple of non-specific defenses that we'll talk about are inflammation and fever. So inflammation, or the inflammatory response, is a local tissue response to injury. So this injury could be an actual physical injury, right? You could hit something, you sprain it, you bump it, and that causes inflammation because you physically damaged the cells. Or it can be because of an infection, right? If you actually have an infectious organism in that area, you're also damaging your cells, and you're going to cause inflammation that way. So anything that kills cells or injures tissues. Again, killing your cells releases all their components, that stimulates inflammation. Anytime you're injuring tissues, that stimulates inflammation. During infection, when we have basophils, they stimulate inflammation. We said neutrophils stimulate inflammation. Mast cells are really important for stimulating inflammation. Again, this is protective. One, it tends to limit the spread of an infection or an area of injury. Think of it as kind of like when you create inflammation around the outside of the area, you're almost walling it off. Right? So you can kind of wall off that area so that it's harder for anything to get out of that area. You're also rushing in a bunch of good protective things, whether it's white blood cells or chemicals to stimulate new blood cell formation or new blood vessel formation or glucose or whatever it is that you need to be rushed into that area. The increase in blood flow is increasing how much of it you can bring to the area. So the cardinal signs and symptoms of inflammation: swelling, right? Because you have an increase in blood flow and vascular permeability. bring more blood to the area, and because you have an increase in permeability, you push more out of the blood. So you've got swelling that occurs. Like we talked about that with edema, right? When you injure an area, how you get edema, you get swelling, you get an accumulation of fluid that occurs because of the increase in blood flow and permeability of the vessel. You get redness and heat, again, because of the increase in blood flow. Your blood is red. Hopefully this is not a surprise. Your blood is red. So if you bring a bunch of blood to an area, it's going to appear red. Also, remember your blood is a little bit higher in temperature than your general body temperature, right? It's like 100.4, and then that heat dissipates when you take your body temp. So the blood is hot. If you bring a bunch of blood to an area, it's going to feel hot. It's going to feel warm to the touch, noticeably warm to the touch. And then pain. It causes pain because we compress nerves and receptors. It's because of the swelling. You bring a bunch of fluid into the area and it literally swells, right? And as it gets bigger and takes up more space, you kind of start to squish everything in that area. And when you compress nerves, that hurts. When you compress pain receptors, that hurts. So you do typically have pain in the area as well.
Is that also why you get itching?
Pardon?
Is that also why you get itching?
The itching is the histamine causes that itching.
Okay.
But yeah, from the receptors, you mean? Yeah, but the histamine is causing it because of stimulating receptors, and that's also, yeah, when causing inflammation itself. And it's like with hives in allergic responses. All right, fever. Fever, again, is one of those things that's kind of annoying. We don't enjoy it, but it is protective. When we look at fever, fever is an abnormal elevation of body temperature, right? So instead of being, whatever, 98.6 degrees, the body temperature goes above about 99 degrees. This, again, is protective. One of the reasons it's protective is because it increases your metabolism. Metabolism, remember, is just chemical reactions. When you add heat to a reaction, it happens faster. Right? We do this a lot. We add heat to reactions to make them occur faster. That's a catalyst. So if you heat up your body temperature, you increase your metabolism. Your chemical reactions go quicker. So that means you can make proteins faster. You can make enzymes faster. You can make things happen quickly. Your cells can divide faster because all of the chemical reactions are occurring more quickly. It also intensifies the effects of interferons. What are interferons important against? Excellent, viruses. Interferons work against or help to stop the spread of viruses. They work better at higher temperatures. Temperature itself, or a fever itself, can also inhibit some pathogens. We have some pathogens that work best at very specific temperatures. So changing the temperature by just a few degrees can interfere with their replication, can interfere with how well they utilize resources, can help to inhibit some viruses and bacteria. So a pyrogen is anything that causes a fever, anything that causes the hypothalamus to raise your body temperature. We'll look at this on the next slide, but remember your hypothalamus is like the thermostat for your body, right? It has your hypothalamic set point. It tells you the temperature that your body should be, and then your body responds accordingly. If your hypothalamus says 102, you're gonna get up to 102. If your hypothalamus says 96, you're gonna cool off to 96. Your hypothalamus tells the body what temperature it should be. So a pyrogen is anything that goes to the hypothalamus and says, okay, we need to heat up. We need to increase our temperature, and it causes some sort of a fever. We have exogenous pyrogens and endogenous pyrogens. Exogenous are like external sources, right? These are pyrogens that come from outside the body and come in and cause a fever. So these could be any type of foreign pathogen. So bacteria, right? When you have a bacterial infection and you get a fever, that's an exogenous pyrogen. When you get a virus, infected with a virus, you get a fever, that's an exogenous pyrogen. Drugs. Some drugs can cause fever. That would be an exogenous pyrogen. Endogenous pyrogens come from within the body, right? Endo means within. So endogenous pyrogens come from our body itself. Our immune system cells, okay, so like our macrophages, once they become activated, our neutrophils, once they come in contact with some abnormal cell, they'll release interleukins, and interleukins will then go up to the hypothalamus and tell it to make prostaglandin, which we'll talk about in a second, and that increases temperature. Okay, so our immune system cells can try and increase our body temperature, and the entry of stuff from outside the body, like bacteria and viruses, can increase our temperature. So lots of different things can act as pyrogens or act as things that cause a fever. So, what do these pyrogens do? We just said that pyrogens cause fever, right? They do this because they raise the hypothalamic set point. They raise the temperature the body thinks it should be. Again, whatever the hypothalamus says, the body does. If the hypothalamus says 98.6, that's what I should be, 98.6. And my body will do whatever I have to remain that temperature. If I get too cold, I'll shiver to warm up to 98.6. If I get above that, I'll sweat to cool off to 98.6. That's the set point. That's what my body's gonna try to be. If I have pyrogens present in my blood and they make their way up to the hypothalamus, the hypothalamus now says, wait a minute, I shouldn't be 98.6, I should be 102. 102 is normal. So your body's gonna do whatever it has to get up to that new set point, that new temperature. The way that that works is that the hypothalamus releases prostaglandins, okay, and the prostaglandins go out to the body and tell the body to warm up. Okay, so just like if you were actually 96 degrees, you would think you were too cold, right? So your new set point is, say, 103, and you're at 98. Your body thinks you're too cold, right? Your body thinks, I need to get up to 103. All these prostaglandins go out to your body and they tell your body how to warm up, right? So how do you warm up? What do? You shiver, right? So you start shivering and that generates heat. You also conserve all of your blood in your core, right? You don't want to lose heat at your skin surface. So these vessels at the skin surface constrict and you hold your blood in your core to keep it warm. That's why when you get a fever, you shiver. And that seems so weird, right? It's like, I'm hot, why am I shivering? Your body temperature says 101, but you're sitting there shivering and it doesn't make any sense. That's because your body thinks you should be even hotter. And so you're trying to warm up to get to that point that your hypothalamus says you should be. Then eventually it breaks and you start sweating, and again, that's when it goes back down again, because all of a sudden the hypothalamus goes back to normal and your body thinks you're too hot. But that's why you kind of go through those stages of shivering and then sweating. It's all because the hypothalamus is changing your set point. So when the body temperature rises, fever is present, and again, that's all protective. That tries to inhibit the microorganisms. It helps to stimulate your metabolism, to stimulate how well your interferons work and how well your immune system cells work. So drugs like aspirin, ibuprofen, Tylenol, or APP is acetaminophen, or Tylenol. These drugs all in different ways, but in one way or another end up inhibiting prostaglandin synthesis. So that's how they decrease fever, right? The hypothalamus heats the body up with prostaglandins. So if you inhibit those prostaglandins from being made, then you inhibit that fever from being stimulated.